And there we go. So today we started on metabolism of triglycerides. This is going to introduce a few new reactions, and then it's going to draw on everything you just learned about glycolysis and the citric acid cycle. This is how your body takes a fat or oil molecule, either one you ingest or one you have synthesized and that it's stored inside you, and turns it into, oxidizes it, breaks it up, oxidizes it to CO2, and extracts ATP, NADH, and FADH2 so that you can get energy for work. And it's a more involved process than just glucose metabolism, but as I said, we're going to draw on all of that. And we'll even show you, how many of you have heard the conclusion that fats and oils are more energy dense than carbohydrates? Yeah? I mean, you can actually look at a food label, you can see how many grams of carbohydrates and then calories per gram. You could do the math, and then how many fats are in the food, and then calories per fat. What is it? Calories per gram of fat or oil? Around 9 kilocalories, yeah. We're going to show you where that number comes from. Well, actually, and some of the math is on your triglyceride metabolism handout. So you should have this in front of you. There's one mistake that was made. It's not huge, but you can fix it with a pen, or you can download the new one fresh. The only thing is here in the section on fatty acid carboxylate to fatty acyl-CoA, I forgot the enzyme name. It's long chain fatty acyl-CoA synthetase, if you want to write that down. I don't really think I'm going to be pushing you on enzyme names with triglyceride metabolism. I'm putting them there for completeness. You'll see that they're mostly sensible. They mostly give you substrate name and what the enzyme does. But just for completeness, if you want to write that down on your handout, or else you can print it out fresh. Okay, so the overall picture of triglyceride metabolism is as follows. This is a triglyceride, and each of the fatty acids have 14 carbons in them. That's just what I chose to save space. A lot of the fats and oils that we consume are in the 16 to 18 carbon range. Recall that this is the fatty acid portion of the molecule. Recall that this is the glycerol portion of the molecule. What kind of bond is this? What kind of functional group is that? That's an ester, which is level 2. Okay. And the first thing you have to do to metabolize a triglyceride is you have to hydrolyze all of those esters. There are 3 ester linkages in the triglyceride. You add water, and then an enzyme called lipase hydrolyzes those bonds. You get glycerol, which is the alcohol portion of the molecule. And then you get 3 fatty acid carboxylates. We've talked about fatty acids before, and these tend to form micelles. They can be soap if you're working with, if you do this reaction with the triglyceride in an iron kettle at really high temperature with lye. But your body uses an enzyme to do this process. And so you get out glycerol and 3 fatty acid carboxylates. So we're going to consider the fate of glycerol and the fatty acid carboxylates separately. Glycerol is going to get modified and then shunted over into glycolysis. So notice that glycerol has 3 carbons. It's a 3-carbon alcohol. And so what we're going to do is use a kinase to transfer a gamma phosphate group from ATP to one of the primary hydroxyls of glycerol to get a phosphoester. Phosphoanhydride in ATP to phosphoester, you expect that to be downhill in energy because that's level 1 in ATP to level 2 in the phosphoester. Second, we're going to use NAD+, to oxidize the secondary hydroxyl of glyceraldehyde, of glycerol. Oh, there's another mistake. Oh, that's so embarrassing. So scribble out the hydrogen there. I need to fix that. Scribble out the hydrogen and put a double bond there. Okay, because that should be dihydroxyacetone phosphate. That should be a ketone. So we oxidize the secondary alcohol to dihydroxyacetone phosphate, which is a ketone. Then, as you recall, that's a material that you can take to step 5 of glycolysis, and you can use triose phosphate isomerase to turn that into glyceraldehyde, and then it just follows through glycolysis and everything else at that point. Okay? I am really, really sorry about that mistake. That's embarrassing. This is what happens when you're tired and in a hurry. Let's see. So we're going to, a little bit later, come back and based on what you already know, draw a reasonable mechanism for those reactions. Yeah?
What was the first one again you said? First mistake?
First mistake was your thing I forgot to write down, or rather I moved it and didn't paste it back in your handout. I fixed it in mine, but it should say long chain fatty acyl CoA synthetase. And the second mistake is this should be a ketone. All right. And that, well, anyway, that's embarrassing. I'll make sure today I update the posted PDF for the triglyceride mechanism handout, so or the triglyceride metabolism handout, so that you'll have a correct copy that you can look at. Okay, then I am recording. Yep. Do we want to start with a prayer? We forgot. We're just forging ahead. Yes. Let's do the prayer after the break, so I don't have to pause the recording. Is that okay? I mean, we could record the prayer. I guess everybody could listen to it. That's fine. All right. I'm not going to have an opening prayer. We're going to have a prayer at midtime saying, this isn't going very well. Could you please bless him to do better? All right. Here's a philosophical question. Did our lack of a prayer at the beginning of the class retroactively cause my mistakes in the metabolism handout? I don't know. Yeah, okay. Well, there we go. So that's the fate of glycerol. It's going to go to glycolysis, and we'll talk about that in a little bit. Next, you've got these 3 fatty acid carboxylates. And again, this is an overview of what happens. We're going to go back and talk about how. The fatty acid carboxylate needs to go from a carboxylate to a thioester. And that's uphill in energy. That's from level 4 to level 1. So we need to pay for that somehow. And the way we pay for that is to take the level 1. I did it again, Leeville. Oh well, we're just going to leave it. We're going to take the level 1 phosphoanhydride and ATP and use it to activate the level 4 carboxylate in the fatty acid. And in the process, we will make the thioester. I'll take you through this mechanism a little bit later. It seems more involved than it actually is. It's pretty simple. But for right now, understand that you need to consume 1 molecule of ATP. Now, one interesting feature here is you don't just make ADP. In fact, the way the mechanism works, you make AMP, which means you clip 2 phosphates off of ATP. When I show you the mechanism, that'll become clearer. But one thing you need to understand about this step is it effectively, because you're hydrolyzing 2 phosphates off of ATP, this step consumes 2 equivalents of ATP. The reason for this is AMP, adenosine monophosphate, that only has the alpha phosphate remaining, your ATP synthase doesn't use that as a starting material. Your ATP synthase in your mitochondria that's powered by the electron transport chain, the starting material is ADP. So if you want to get AMP and recycle it, what you have to do is take AMP, phosphorylate it with ATP to get 2 molecules of ADP, which you can then take to the ATP synthase in mitochondria. So every time you go through this reaction of activating the fatty acid carboxylate, you're effectively consuming 2 equivalents of ATP. One here, and then the second there. Questions about that? Where does the second one come from? The fact is that in order to recycle AMP, you first have to convert AMP to ADP, and that requires a second molecule of ATP. And the other way to think about it is just in going from ATP to AMP, you've hydrolyzed 2 phosphates. You've broken 2 phosphoanhydride bonds. So that counts as basically 2 ATPs. Yeah. Okay, other questions? That's not hugely important except for when we come back and figure out exactly how much ATP we're going to get out of a triglyceride. We're already down 2. Yeah. Okay. So at this stage, what we're going to do is just pause and look at the energy balance or rather look at the ATP output of glycerol. I guess I should have paused here before we went on to the fatty acid carboxylate. This is in your handout. I got these numbers just by reviewing the handouts of glycolysis and the citric acid cycle, and then what I showed you for glycerol. And this is a good way to review how well you understand metabolism. And you may want to go through, not just taking my word for it, but go through and do this by hand yourself. For example, the reaction I showed you above, glycerol goes to dihydroxyacetone phosphate. In that process, we're down 1 molecule of ATP, but we make 1 molecule of NADH. Okay? Then I went back to the glycolysis handout, and I noticed that starting with dihydroxyacetone phosphate, as you go from there to pyruvate, you're going to make 2 ATPs and get 1 NADH. Now that's different from the overall balance from glycolysis, because glycolysis starts with glucose. We're just starting with dihydroxyacetone phosphate here. Then from pyruvate, we go through the pyruvate dehydrogenase complex to get CO2, acetyl-CoA, and 1 NADH. And then acetyl-CoA goes through the citric acid cycle, gives you 3 NADHs, 1 FADH2, 1 GTP, and 2 CO2. So if you add all of this together, the process of glycerol going to 3 CO2s generates 2 ATP minus 1 ATP, so that's just 1 ATP. 1, 2, 3, 6 NADHs, 1 FADH2, 1 GTP, and with these assumptions that we've already talked about, that's 18.5 ATP molecules. Some of you are going to be like, well, how can you have a half ATP molecule? You can't. These are approximations. So somewhere between 18 and 19 ATP molecules for a glycerol. Okay? So that's just the 3-carbon glycerol portion. Now any questions about that? I think the horror story you're wondering in your brain is, at some future exam, am I going to show you a triglyceride and ask you, how many ATPs do I get from that? And are you going to have to know? I haven't decided yet. I'm probably going to put it on problem set 6 though, because I think it's worth going through to help review what you've done. Okay? At least, I am a villain, but at least I'm monologuing and disclosing all my evil secrets so that you can prepare. Okay. let's talk about, all right, the fatty acid carboxylate. We've already done activation to the thioester. We're down 2 ATP molecules for going from the carboxylate to the acyl-CoA. What happens next is a really beautiful and simple process called fatty acid beta oxidation. And you'll see why by focusing in on what happens to the beta carbon. So that's the carbonyl carbon, that's the alpha carbon, that's the beta carbon. And notice that in the fatty acyl-CoA, the alpha and beta carbons are saturated. That is, there's no pi bonds, just a bunch of CH bonds. So the first step is a dehydrogenase. So again, the names you can sort of tell what's going to happen. Acyl-CoA is the substrate, the molecule that it acts on, and dehydrogenase tells you it's an oxidizing enzyme. The other reason you can tell is because you go from FAD to FADH2. And recall, as we learned last time, FAD is a hydride acceptor and a proton acceptor. We learned about that in the oxidation of succinate to fumarate. And this mechanism is basically exactly the same as that. In this mechanism, nature's going to remove a proton from the alpha carbon and then kick off a hydride from the beta carbon. And that's going to give you this trans-enoyl-CoA, where you now have a double bond between alpha and beta carbons. It'll be useful to you if you can connect this to the reaction of succinate to fumarate in the citric acid cycle. We'll go through this mechanism, probably not today, probably on Monday, but when we do, you'll see it's basically the same. So seeing connections to things you've seen before will be important. Next, just as we did, this next step is going to be like fumarate. That's a terrible penmanship. Fumarate to malate in the citric acid cycle. The enoyl-CoA is an alpha-beta unsaturated carbonyl compound, and the beta carbon is electron poor. And so what you're going to do is add water to the beta carbon as a nucleophile, and then you'll pick up a proton on the alpha carbon through some enzyme, acidic enzyme side chain. And that installs an OH group at the beta carbon. In the next reaction, you're going to oxidize that beta OH group from the alcohol to the ketone. And you can tell it's an oxidizing reaction because the enzyme name is hydrogenase. And of course, along the way, you're going to make NADH. NAD plus is a hydride acceptor. So you'll expect this mechanism to be very similar to other NAD plus mediated oxidations. That is, you expect a base to remove the proton from the oxygen, and electrons will kick down and remove the hydride to be received by NAD plus. Then the last step is really where the magic happens, and we'll need to go through this mechanism in detail because I don't think you've seen it before. The mechanism, we're going to call it a retro-Claisen reaction, and it's similar to a retro-aldol reaction, but the effect is going to be to clip off the 2-carbon unit that is composed of the carbonyl carbon and the alpha carbon. and then the beta carbon here becomes the new carbonyl carbon here. So we're going to break the bond in this step between alpha and beta carbons. We're going to clip off a 2-carbon unit of acetyl-CoA that's going to go to the citric acid cycle. And then we'll take our 12-carbon unit and go straight back to the beginning. And the enzymes that do this form sort of a large complex, and things get rotated around until there's nothing left of the of the fatty acyl-CoA. You've just trimmed it into acetyl-CoA units. Okay, we'll go through each of those steps individually. I just wanted you to see the overview. Notice that the first step is an oxidation where we lose H2 from the molecule, and we lose one from the beta carbon and one from the alpha carbon. And then in a subsequent step, we put an oxygen on the beta carbon, we oxidize it to the ketone, and then the retroclason reaction happens where we clip off a 2-carbon acetyl-CoA unit. Questions about that? Yeah?
Just for context, where do the cells sustain this?
Ooh, I don't know that. Somebody needs to Google that. I should know that. Off the top of the head, where does fatty acid beta oxidation happen? I think mitochondria.
Let's find out.
Thank you. Thank you for Googling that for me. You'd think I would have done that before class, but.
Right.
Ding, 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 ding.
Extra credit.
Negative extra credit for you because I was right? Is that? Oh, okay, yeah. Sorry. Maybe that will make up for my earlier mistake, right? I get at least, okay. Earlier mistake, forgiveness. So yeah, fatty acid beta oxidation happens in mitochondria, which I guess makes sense because if you're going to be churning off acetyl-CoA, then it makes sense to have it going right where the citric acid cycle is. Okay, here's an interesting question. How many acetyl-CoA units can I get from a 14-carbon fatty acyl-CoA? 2, 4, 6, 8, 10, 12, 14. Okay, so I can get 7 acetyl-CoAs. Here's a question. How many times through beta oxidation are required to clip that up into 7 acetyl-CoAs? You got to think carefully here because you want to say 7 because there's 7 acetyl-CoAs, but it's not. 6, right? The reason for that is you've only got 6 bonds to break, and importantly, the last time through, your product is going to look like this. Sorry, your final starting material is going to look like this, and the truncated acyl-CoA thioester at this point is just going to be another molecule of acetyl-CoA. So for a 14 carbon, the first you go down to 12, second to 10, third to 8, fourth to 6, fifth to 4, and then the 6th time you break 4 into 2, and then they're both acetyl-CoA. So that's where people get tripped up in sometimes in doing the math for just how many acetyl-CoAs and how many times through beta oxidation do I need to go. Just remember, it's the number of carbons in your fatty acid divided by 2, and then you got to subtract 1 because the last time through you make 2 acetyl-CoAs. Questions about that? That sort of makes sense? Okay. So let's look at the energy output or the ATP output from a 14 carbon carboxylate. In the long chain fatty acyl-CoA synthetase, we're going to turn the carboxylate into the CoA thioester. That takes us down 2 ATPs. Then, through beta oxidation, we convert that 14 carbon fatty acyl CoA into 7 acetyl CoAs. Each round of beta oxidation, if you look up here, gives you 1 FADH2 and 1 NADH. And since this 14 carbon acyl CoA goes through 6 rounds of beta oxidation, we get 6 NADHs and 6 FADH2s. With me so far? Just sort of counting up the total. Then we need to take each of those 7 acetyl CoAs to the citric acid cycle. So here's where you got to look up exactly what the yield from the citric acid cycle is. It's per acetyl CoA, it's 3 NADHs, 1 FADH2, and 1 GTP. So I just scaled those numbers up for 7 acetyl CoAs. So add it all up, 1 14 carbon fatty acid carboxylate gives you 14 CO2s, negative 2 ATPs, 27 NADHs, 13 FADH2s, and 7 GTPs. With these assumptions about how much each one is worth, you can come up with the number if you do the arithmetic, and you don't have to do that in your head right now, 92. Yeah?
Is this explaining the molecules that are shown above? I guess I'm just kind of confused on what all this is trying to explain besides how much ATP you get.
How much ATP you get from a 14 carbon carboxylate is what I was trying to explain. The stuff above shows you the reactions that are required for that. So this fatty acid carboxylate to fatty acyl CoA, this first line, is represented by this reaction, which in retrospect I probably should have put on the next page. Man, maybe I'll revise it again. And then this line, what happens to the 14 carbon fatty acyl CoA is summarized here, but you got to go around 6 times for it to happen. And then the last line describes the fate of each of those 7 molecules of acetyl CoA that you make. And to get that, you need to review what the citric acid cycle does. So we're following a carboxylate all the way through beta oxidation 6 times, then to acetyl CoA, and then we're following those 7 acetyl CoAs that we make through the citric acid cycle. And we're summing up all of the NADHs that we get out, and all the FADHs that we get out along the way. Yeah?
So does this all start with the glycolysis cycle?
For the fatty acid, no. Because glycolysis is how you break sugar or glucose into pyruvate and then acetyl CoA. And the fatty acid just goes straight to acetyl CoA. The one time we bring up glycolysis is for what happens to the 3 carbon glycerol. That bit from the triglyceride, we'd already talked about that, but that bit from the triglyceride gets turned into dihydroxyacetone phosphate, and then that goes through the remaining steps of glycolysis. So yeah, it's weird because you got this triglyceride of this portion, the glycerol goes over here to glycolysis, the fatty acids go over here to beta oxidation.
So we were showed that from starting from glycolysis, you can go straight into this citric acid cycle because it starts, well, it goes into acetyl CoA.
Right.
So does that mean that the triglyceride metabolism is something completely different from glycolysis as the citric acid cycle?
So I guess what I would say is triglyceride, they're overlapping. It's like a Venn diagram that overlaps, but not completely. So, so if you think about glucose metabolism, and triglyceride metabolism, there's a few spots of overlap. One is the citric acid cycle. And then the other is part of glycolysis because of glycerol. But steps, I would say 1 through 4 are unique to glucose metabolism. Triglycerides don't take advantage of that. And then beta oxidation, the activity of the lipase enzyme to hydrolyze the carboxylates off, that's separate. But you're right, they do funnel together into the citric acid cycle. Yeah?
So these are all like different types of metabolism, just different ways that different molecules are turned
Yep.
into ATP.
Different ways that different molecules are processed, oxidized, ultimately converted to CO2, and along the way we get ATP.
This isn't super important. I'm just wondering, like, are triglycerides stored like in adipose tissue? Are they stored as triglycerides, or
Uh-huh.
are they stored as something else?
Yeah, in adipose tissue, they're stored as like fatty deposits in adipocytes, or the fat-containing cells. Yep. Go ahead.
So with this, are we just kind of, because we almost do like a hydrolysis or in like catalytic red,
Yeah.
and then it breaks into two.
Yep.
Are we just focusing on one at a time?
Yes.
And with the dihydroxyacetone phosphate, are we just done with that, and then we just work with the fatty acid carboxylate until it?
Yeah, well, so done with it, no. You're right that you get to the dihydroxyacetone phosphate, and then I'm saying that gets processed, that can be processed in the 5th step of glycolysis. So you'd have to go back to your glycolysis handout and see what happens to dihydroxyacetone phosphate.
So that wouldn't be an end product.
So no, that's not an end product. To do that, maybe, how many days ago did we even do glycolysis? Let's see. Not that many days ago. Boy, what a week, huh? Yeah, so this is dihydroxyacetone phosphate. That comes here and then goes all the way here and then all the way there to acetyl-CoA.
So you can produce it. Wait. So does that one come from the triglyceride metabolism, or does it also get produced through glycolysis?
Yes, both. So dihydroxyacetone phosphate is a product of glycolysis, but the enzyme that processes dihydroxyacetone phosphate doesn't care where it comes from. So you can supply this, and now we need to amend our little diagram. You can supply this from glycerol metabolism. Yeah. And so, I mean, if you want to look this up, all metabolic processes are interwoven in a network like this. If you want to see it, biochemists have worked out a lot of them. There's a big, enormous poster, and it would probably take up all of this wall, and it would be pretty small print, and it would blow your mind. In the break, I'll look it up, and we'll try to look at it just for fun, and then we'll stop worrying about it, but it's pretty crazy. And the cell balances, all of those based on what you need to do at a given time. Okay, good questions. Let's see, where were we there? Yep, so that's we do the lipase reaction, we deal with glycerol, that then can go to glycolysis and be processed. We do the fatty acid carboxylate, that gets modified to the thioester, then goes through beta oxidation to become acetyl-CoA. What we summarized here was the ATP output from glycerol, which is 18.5 ATPs. That's following through the processing of glycerol, then dihydroxyacetone phosphate to pyruvate, and so on. And we were just getting through the conclusion that for each fatty acid carboxylate, once you go through beta oxidation and go through the citric acid cycle, 92 molecules of ATP. Okay, you can get that just by following all the reactions through. Other questions about the overall view of things? So one triglyceride, in one triglyceride you have one molecule of glycerol and 3 14 carbon fatty acids. So that ends up being 14 times 3, 42, 45 carbons total. If we do all of this from lipase to glycerol metabolism through the remaining portion of glycolysis, we'll call that glycolysis light because it's just the last few, and then through fatty acid beta oxidation, and then through the citric acid cycle, you ultimately make 45 molecules of CO2, 18.5 ATP from glycerol, and 92 ATPs from each of 3 14 carbon fatty acids for a whopping 294 to 295 ATPs from one triglyceride. Now remember, just for comparison, one glucose, which is 6 carbons, equals 32 ATP. So molecule per molecule, triglycerides lead to way more ATP. Yeah?
So biologically, like, our brains and stuff use majority glucose, but like, it seems way more efficient when something goes wrong to like, really operate from like, beginning with triglycerides, because obviously like, the ATP output is so much lower. Why are we like,
So this, yeah, it's an interesting question. The yield of molecule per molecule, the yield of ATP is much higher. Though whenever you're comparing two things, it's important to compare apples to apples and so on. By the way, I hate that game. Ugh. Yes, we'll have a discussion about that a little bit later. But anyway, per molecule, you're right, it's dramatic. Although this one has way more carbons. So we could do it per carbon as an idea.
and that depends as well, but like, why are we not, do we just not have, we do both, but like,
So I looked it up, and your brain does use mostly glucose. It's not that your brain cells, and there's a lot of different kinds of cells in your brain, but it's not that they can't do fatty acid beta oxidation, it's just it's not as efficient for them.
why are we starting from triglyceride? Like, why is it, you know what I mean? And then like breaking it down to get insulin.
Why doesn't your brain start from triglycerides?
Do we, I guess, like, what process happens more often? Like, are we typically operating from like using the triglyceride? Like, do we use mainly triglyceride, or do we use mainly glucose? I feel like my question now sounds stupid.
No, it's a great question, and in some ways you're probably asking things that I'm not necessarily qualified to answer. If I'm guessing a little bit, it would be how fast you can get ATP versus just what's, I mean, evolutionarily, the idea is, I think we'll see that triglyceride storage, we need to do just a little bit more math to show that triglycerides are indeed more energy rich and energy dense than glucose is per gram. And so that means it's more efficient storage. It doesn't make sense to have, you could store big sacks of glucose inside you, but it wouldn't pack as much energy. So you're lugging around more mass to have more storage. So more energy dense storage makes sense, requires less investment of energy to just move around. Storage though, because it's so energy rich, it's valuable, so you probably don't want to tap into it unless you really need to. And so, and if you can get plenty of carbohydrates in your diet, then it's not such a big deal to be running off glucose. Yeah, but that's just guessing, and there's probably a whole lot more to it. As to why your brain doesn't use a triglyceride to begin with, I hadn't even thought about getting triglycerides across the blood-brain barrier. That's probably hard. I don't know, right? So they can do fatty acid beta oxidation, and they do, just not as efficiently as muscle cells and others. Yeah?
Do unsaturated fatty acids do beta oxidation the same way
That's a fantastic question. So unsaturated fatty acids have double bonds and do they do it the same way? There's a little bit more processing that has to happen, and I don't know what it is. It's a good question. Presumably, you've got to, if the double bond's in the right place, you'd be like, sweet, only it needs to be trans instead of cis, so there's at least an isomerization step. There may also need to be a move the double bond over one step. Trying to remember the details, but I got nothing. Yep. Okay. So should we do just a little bit of math? A simple way would just be to divide the number of ATPs by the number of carbons. So let's pull out our calculator because I didn't do this ahead of time. I did some other math ahead of time. But let's see, 32 carbons divided by 6. Some of you can do that in your head, but I certainly can't. 5.3 ATPs per carbon. Whereas the triglyceride, 294.5 divided by 45 equals 6. So per carbon, it's just a little bit more efficient. If we get to thinking about mass, it's quite a bit more efficient. So if you take, there was a simpler way to do all this math, but the numbers I brought were related to the questions that we talked about last time. So if you recall, last time we said that average human metabolic needs per day was around 2,000 kilocalories. And we said that ATP hydrolysis, the delta G for that is anywhere from -7 to -15 kilocalories per mole. So we used that to calculate that you needed about 130 to 300 moles of ATP per day, that you're going to consume that much. And we figured out exactly how much ATP you have in your body, and therefore how many times an ATP molecule needs to be recycled. The number was like 1,000 to 2,000 times, and you thought you were busy. But we also translated that based on the idea that 1 glucose equals 32 ATP. We said that you needed about 0.7 to 1.7 kilograms of glucose per day if that's all you were eating. Now, we've just said that for one molecule of a triglyceride with 3 14 carbon fatty acids, that equals 294.5 ATP. And if you do some assumptions about the molecular weight of if you calculate the molecular weight of the triglyceride that I've shown you above and do the math, it ends up being 0.32 kilograms, 0.32 to 0.74 kilograms of the triglyceride. So you can divide these numbers by these numbers and say that per gram, the triglyceride yields about 2.3 times as much ATP as glucose. So that's efficient energy storage. This is why you don't want to be packing sugar around when you can pack, you want to be packing, this is why you store glycogen in your liver for easy usage, but for long-term usage that you aren't going to tap into that frequently, but you want it in case you no longer have access to glucose, you're going to use triglycerides. All right, and probably if you go to a food label and do gram per calorie for carbohydrates versus gram per calorie for fats and oils, probably you, and compare the two numbers, probably you come up with something close to 2.5 to 2. Yeah.
Will there be like a question on the test like this, and if so, what would it look like?
Ah, thank you for phrasing it that way. Instead of is there, would there, could there be? Yeah, anyway, would there be a question on the test about this? I just did this math today because I think it's cool, so I haven't really yet decided. I mean, some of you are in food science, nutrition, dietetics, this kind of thing is important to you. Others of you are hoping to be nurses someday, and still nutrition is an important aspect of things. I'm less really concerned with can you do arithmetic, but I'm worried if I say no, this isn't going to show up, then you're never going to pay attention to it again, and I think it's important. So I will try to figure out how I'd ask a question about this, and I'm going to put it on problem set 6 so you can see. I'm sorry I have to punt on that. I mean, I could really draw a triglyceride structure and say how many molecules of ATP do I get from this. What I'm not going to make you do is calculate molecular weight of a triglyceride and then do grams per mole and all of those kinds of conversions. That's general chemistry stuff, and we don't have time for that. Yes?
This is just like a curiosity question, but so you said that like if all you were eating was glucose, you'd have to have like 0.7 kilograms
Yeah.
per day. What happens, I guess, in this like process when like people are like not eating, like when they're like starving?
Oh, that's a fascinating question. What happens when you're fasting? Couple of things first, I don't know. I mean, I sort of know because we've talked about some of this. If you're not taking in glucose, your muscles and liver can run off glycogen for a while. You liberate glucose from glycogen. I showed you the structure of glycogen, and there are enzymes that clip off the glucose molecules, and so that can supply needs for the cells and up your blood glucose level to a point. Once that runs out, your body starts to do something called gluconeogenesis, where it starts making pyruvate and going up backwards through the glycolysis. And there's a few different enzymes that are needed to do this, but so your body can make glucose to supply the blood and the brain. How's it going to do that? Well, you need to start breaking down triglycerides into acetyl-CoA. And then you can use that as the starting point for some other stuff. Along the way, there are people that talk about ketone bodies. Those are related to the intermediates of fatty acid metabolism. Notice that after this oxidation step, you have a ketone. And so I don't know how those are processed. I know they can be detected in your blood and even on your breath. If you're severely fasting and your blood glucose is starting to get low. And your body can use those. I don't know any more about it than that. Okay, what else? Also, incidentally, this kind of math is what's used when somebody tells you if you want to lose a pound of fat per week, you need a calorie deficit of this per day. This is where those numbers come from. Because we've just described how much ATP you get per kilogram of triglyceride. So, and ultimately per calorie that you want to use per day. So you could use these numbers to get that conclusion. Yeah?
You said earlier that we're going to go further into the triglyceride metabolism and
Mechanisms? Yeah. Yep, so this is just the overview. Sometimes it's helpful to see an overview of what before we delve into the details of how. But the details of how are really awesome. So let's take a break now, and we'll remember to pray after the break. And then we'll see how things go with the mechanism.